Hello everyone. So today we will learn about proteins. So one Dutch chemist named G.J. Mulder first observed one heat coagulable nitrogenous compound in milk. Now these heat coagulable nitrogenous compounds are proteins, and he observed casein protein that is found in milk. Proteins are derived from the word proteose, meaning holding the first place, or we can say or preeminent, or we can say primary as well. So this was the discovery history of protein. Now proteins are polymerization of amino acids. So number of amino acids are linked through peptide bonds. Now these proteins contain carbon. If we talk about the ratios, 50% carbon is there, 7% hydrogen is there, 20% oxygen is there, 16% nitrogen is there, sulfur is 0 to 4%. So these are the major compounds that are present in proteins, but some contains, that means some proteins also contain zinc, phosphorus, copper, iron, manganese, molybdenum, etc. So some proteins contain these, but not all. But these compounds, carbon, hydrogen, oxygen, nitrogen, and sulfur, is present in all proteins. Now, proteins are, we are talking about the properties. These are the fundamental units. Of cell, or you can say fundamental structure also. Second, they are the biological and physiological functions are performed by proteins. Biological and physiological, like they act as enzyme. All the enzymes that are present in the body are proteinaceous in nature. They act as hormones. They act as blood clotting factor. Like fibrinogen. They act as oxygen carrier, like hemoglobin. Ionic pumps, like potassium pumps, sodium pumps. They act as ion channels. All the channels that are present, like voltage gated channels and all, they also act as immunoglobulins, as membrane receptors, lipid carriers, electron carrier, contractile machinery of muscles. So unbranched polymers linked through L alpha amino acids, and they covalently linked through peptide bonds. So proteins are always L alpha amino acids will be a protein, not D. So L alpha amino acids having unbranched polymers, they covalently linked through peptide bonds to form proteins. And if more than one PP chain, that is polypeptide chain, is present, the name is oligomeric or multimeric. Example for oligomeric or multimeric is CPK, insulin, LDH, HB, ALA, dehydrogenase. Now proteins on hydrolysis gives out mixture of free alpha amino acids. If we see the structure of protein, COOH, one amino acid, and amine group of any other amino acid is bonded. This terminal containing COOH is known as the C terminal, and this terminal containing NH2 known as N terminal. So this one is known as alpha amino group, and this is known as alpha carboxyl group. So alpha carboxyl group contains CO bond. So this here CO bond is takes part in this formation, not double bond O, C single bond OH. So this H is removed, and this valency of O becomes free, and this valency later makes bond with NH2. Here also H is removed. So if we see the reaction, this is carboxyl group of C terminal OH, and here one amino acid is present, and similarly here one NH2 is present, and one another amino acid, say amino acid 2, and this one is amino acid 1. That means there are two amino acids takes part in this. So this OH and this H will be removed. So minus water, that means water is removed, and now this becomes C double bond O N H amino acid second one, and here amino acid first one. And as we have read that this is known as peptide bond. So in this video we have learned about the proteins. Next we will learn about the classifications of proteins on the basis of several factors. So thank you.